Thank you. Welcome to the week 3 of the Frontier Mechanical Engineering Seminar Series. I would like to go ahead and welcome all of you on behalf of the organizing committee. And just for a brief, I'd like to welcome 3 new members on the committee from Princeton, Notre Dame, and UT Austin. Just to give a highlight, we started last semester this series to bring junior faculty member in pairs to talk about the research. And in this semester, we have added a new component where we are bringing distinguished speaker as a part of the series. And for the next 2 weeks in a row, we will have 2 speakers, one from Georgia Tech, which we will speak today, and the next one is from UCLA. And after that, we're going to go back to our normal schedule, which is 2 speakers from from 2 schools, which are partnering on a thematic area. So with that, let me just give a plug for next week, April 2nd. I know some of you have a spring break, so maybe you can watch it from the beach. So we will have a seminar in data science area, and the speaker will be Dean Jayati Murthy from UCLA. And the moderator for that session will be Professor Yogi Joshi from Georgia Tech. And now let's start. For today, our theme is bioengineering, and it's my great pleasure to have our very own Andrés García, but I will not introduce him. I will go ahead and introduce a friend of mine who's going to do the moderator for today. Professor Joanna Temenofov. She is a faculty member in the School of Biomedical Engineering, which is a joint department between Georgia Tech and Emory. She holds the Carol and David D. Flanagan Professorship in this school. She's also their Associate Chair for Translation Research, and she's also the co-director of the Center for Manufacturing, which we have cell manufacturing NSF ERC. Along with that, she has several other service activity on campus. I don't know how she gets her time, but when she's not doing all her service, she is an amazing researcher in the tissue engineering area. And she has been recognized worldwide for different contribution, including she is now a fellow of different society. I'll just name a couple of them because the list is long, which is the Biomedical Engineering Society. She's also a fellow of American Institute of Medical and Biological Engineers. And I had a few other things on the list, but I would really like Joanna to be here because over the last 1 year, we have worked on some of the COVID response task force together. She knows Andrés for a long time, so I could not find a better person to introduce Andrés. With that, Joanna, thank you for your time, and it's all yours.
Well, thank you very much, Devesh. Yes, it may be to Andrés' detriment. I may know these stories, but we'll keep it above board for this introduction. But I guess I wanted to start a little bit. Devesh had asked me to talk a little bit about bioengineering, and I kind of think this is one of those things that needs very little introduction at this point. It's kind of been at the forefront of our lives for a year or so, but it is the application of engineering principles to biology, discovery of new biological principles, and also for human health. And so I don't think I need to explain how traditional engineering approaches have been used in the past year. And we've, Devesh and I and Andrés have all been on this response task force at Georgia Tech, and we've all talked about everything from, how to, new delivery vehicles for vaccines, supply chain, even modeling of airflow in various buildings. And so all of these things for the past year are really driving our ability to determine if our surroundings are safe and to make human health better. And so I think that that's been what's really drawn me to the field over the years is the potential for immediate impact and making people feel safer and able to move without pain and to be free from disease. And I think that that's really kind of come to the forefront in the past year. So that's the best of bioengineering. One of the things that, as we've been on this response task force, I think that has come to my attention is as we've brought more and more people on campus, engineers and scientists, to look at these biological problems, what's surprised many of them is actually that, you know, with great promise comes great responsibility. So one of the things I just wanted to point out that continues to be a challenge for bioengineering is kind of threading that line between innovation and making sure that our innovations are very safe and passing all of those hurdles, which eventually end in FDA approval. And so I think this is a unique challenge for the field as opposed to some other engineering fields, and really needs to dominate our thinking from very early on when we're talking about bioinnovations innovations in bioengineering. And the reason I bring this up is because I really think Andrés is an exemplar of someone who's tried to thread that needle and has done that very successfully over the course of his career. So as was mentioned, I have been lucky enough to know Andrés for over 16 years since I joined the faculty at Georgia Tech, and I'm very happy to call him a mentor and a friend. Very excited to see all of the great things that he's been doing before and since I've joined the faculty. His research program is actually in combining material science and some cutting edge biological techniques to learn more about mechanobiology, but also to develop biomaterials that have applications in the healthcare field. And hopefully he'll be giving you some examples of that. He is a Regents professor in mechanical engineering at Georgia Tech. He also, in his free time, directs our Petit Institute for Bioengineering and Bioscience, which is a unique interdisciplinary institute that really brings together engineers and scientists, and has a lot of core facilities as well as a lot of programming that really drives all of our research to the next level. So we're really grateful for his leadership there. In terms of his stellar research career, he's also received too many awards to list all, but just as a highlight, he started with an NSF Career Award. He's won multiple awards from our society, the Society for Biomaterials. He also acted as president a couple years back. He is a fellow of the International Union of Biomaterials Science. He's a AAAS fellow, ASME, AIMBE, and probably the most exciting in the past year, he's been elected a member of the National Academy of Inventors and Engineers. And so we're really excited and proud for him for that. In addition to all of this great research and these accolades that are so well deserved, I really want to acknowledge Andrés for always being a champion for entrepreneurship. He is a co-founder of 3 startup companies based on his technologies, but he's also really tried as director of the Petit Institute to bring more entrepreneurship academies and speakers in to help people that are really starting to navigate this idea of how do we commercialize and how do we innovate in the bioengineering space. So with all of that, I'm going to now turn it over to Andrés, and he's going to tell us more about his work in bioengineered synthetic hydrogels for regenerative medicine.
Thank you so much, Jonah. That was wonderful, and it's always great to have a dear friend introduce you, and I appreciate you coming over from BME to do it. First of all, I hope all of you and your loved ones are safe, and hopefully you'll get the vaccine soon.
You're on mute, Andrés.
Muting myself. Somebody unmuted me. Can you hear me?
Hey, you're fine now.
Yeah. And we can see the slides? No slides. Huh. Devesh, I think this is all your fault.
You have to go from slideshow mode. Just remove the slideshow mode and come back. It will share.
Okay, let's try. We had checked this with Ben before. It's good. I'm at the Georgia Institute of Technology. Try thing. Okay. Do it, I hope. Did that work?
Yes.
Yes.
Okay, great. So again, thanks also to Devesh and the committee for inviting me. Let's see. Okay, some required disclosures. So today I will describe to you our work on engineering hydrogels for controlled delivery of therapeutics. And for therapeutics, we're primarily interested in delivering proteins and cells. Many of you are familiar with hydrogels. Two everyday examples are going to be Jello, right, and snot. And that's my son's favorite hydrogel of interest. So as you know, or as many of you are aware, there are many different types of hydrogels. They can be of natural origin, and you can consider most of your tissues to be of that kind. And they also can be of synthetic source. So our work has centered on synthetic platforms. We work with primarily a polymer called polyethylene glycol, history of use in biomedical devices. And our specific contribution to the field was on exploiting this particular chemistry, the maleimide chemistry, to generate the hydrogels that I will talk about. And hydrogels are water-swollen polymer networks. So the systems that I will describe today are about 95% water, so 5% is really the polymer component. And over the years, we have shown that this PEG 4-mile system really serves as a flexible delivery platform for therapeutics. There are several advantages of this platform over other natural and synthetic hydrogel, including other PEG hydrogels. And these include a well-defined structure that allows us to model these materials using hydrogel theory. As I mentioned, a key aspect is the cross-linking reaction, and we have shown that the maleimide thiol reaction really has minimal toxicity. And this is important where applications using sensitive cells such as stem cells and pancreas. An important attribute is that we have metric incorporation of biomolecules. And this is important because the PEG polymer itself does not have any combination sequences for cells. So cells cannot interact directly with the material. And what this allows us is to incorporate different biological signals into the network with very high precision. And this is illustrated by the plot here, in which we're incorporating an adhesive peptide, RGD. So this is a peptide that promotes cell adhesion. And you can see that as we plot the input concentration and how much is tethered in the backbone, we get complete and stoichiometric incorporation of those signals within the material. This allows us to precisely engineer different aspects into the hydrogel. We also have control over the polymerization kinetics. I will show you in a second how we exploit that. And then finally, we have shown that when we make the systems to be degradable, the products elicit no local or systemic toxicity and minimal inflammation, and they are secreted via the urine rapidly within 24 hours. And these are important considerations if we ever going to apply the systems to be used in humans. So the basic strategy is we start with a 4-arm PEG macromer that is N-functionalized with the maleimide groups. As I mentioned, we often incorporate signaling peptides and molecules. So we react those to generate functionalized precursor. And then in the presence of a dithiolated cross-linker, and we can add cells or protein at this stage, the precursor is cross-linked into a network. Because we have complete control of this synthetic system, we can add cross-linkers that can be cleaved and degraded by specific proteases, which are enzymes that could be in the healing environment. So in the presence of those enzymes, the cross-links get caught, and you go from the network to the small degradation products. So over the years, we have shown that this system affords precise control over the biophysical and biochemical properties of the network. And we can independently tune the mechanical properties, the type of adhesive peptide and the density, the degradation sensitivity. This is really a very modular system. As I mentioned, we can manipulate the cross-linking reaction And what that allows us to do by controlling the kinetics is we can control the format of the hydrogel. So for example, we can cast and generate bulk implants in the lab, and those we can use for studies of stem cell niches and organoids, and I'll show you some data on that. Or we can take those bulk implants and put them in vivo inside animals. Great responses. So we've engineered materials that release in a controlled fashion a very potent vasculogenic factor. You can see the release profile here. And that promotes very potent and stable blood vessels to form in vivo. And you can see this beautiful three-dimensional reconstruction of the implant and the blood vessels that are infiltrating within the implant. This is all inside the animal. Again, controlling the polymerization kinetics, we can formulate the material as an injectable carrier in which we have liquid precursors that then get mixed at the site of delivery where they gel and solidify. And using that, we can use catheters to deliver them to different sites. So for example, in this application, we have delivered liquid precursors directly into the heart of a pig, in this case, using human-sized catheters. And finally, we can use microfluidic polymerization to generate hydrogel drops or particles instead of a bulk gel. And we can use those to deliver proteins and even encapsulate cells with high viability. What I will do today is I will describe 3 applications so you have a flavor on how we can engineer the material for the particular application of interest. The first case deals with treating type 1 diabetes. Many of you know patients of type 1 diabetes. About 1 in every 80 children in the United States is a type 1 diabetic. So in type 1 diabetes, the patient develops an autoimmune response to the cells in the pancreas that secrete insulin. So those cells are called beta cells. And essentially, these patients lose the ability to secrete insulin. That results in high levels of glucose, which are toxic to the patient. So one of the major production of insulin, and many of you know that that's how type 1 diabetics manage their condition is with insulin injections. Now that's management of the disease, that's not cure of the disease. So to date, the only cure of diabetes has been islet transplantation. And in this procedure, what you do is you take islets from a cadaver and you reinfuse them into the donor. Okay, and the idea is that you're restoring the cells that provide insulin in response to the glucose level. And these results here really show that, and I really love the results. So what we're showing here are the blood glucose levels in one patient for 4 different days. So each trace is a separate day. Anything inside this gray zone is the normal blood glucose level. Anything above is hyperglycemic, right? Anything below is hypoglycemic. And you can see that this patient throughout the day, so he's fitted with a continuous glucose sensor. You can see that this patient has wild in glucose levels, right? So there's times that he is hyperglycemic, which is not good long-term. And more concerning, he has episodes in which he becomes hypoglycemic. And those, this can lead to sudden death. Okay, so this is a patient before islet transplantation. This is a patient after islet transplantation. And you can see the remarkable control that the patient has over time. So, I mean, this is really a cure for these patients. So, while islet transplantation is an effective cure, it is not durable. And that means that most of those patients, after 3 to 4 years, lose this control and then have to go back on exogenous insulin. So there are significant limitations why islet transplantation has not reached broad applicability in patients. The one that I will discuss how we address is really one of the major limitations, which is immune rejection to the graft. So the patients can we engineer a biomaterial that will induce the patient to accept the graft without using chronic immunosuppression? And this is a major goal of the field for 2 decades. So our efforts have focused on a very potent immunomodulatory protein, which is called need to understand is that this protein, all of you, we all have it, and it's a very important protein to maintain immune tolerance in the body. Okay, so just as a way of background, we know that activated T cells, which are the cells that would end up rejecting the grafted tissue, express the receptor for Fas ligand, which is called Fas, and become sensitive to Fas ligand apoptosis. And this is programmed cell death. So by expressing Fas ligand, your body is able to get rid of the T cells that want to kill whatever target cells. Okay, so the idea is, can we use this system of Fas ligand to induce, to kill the T cells to prevent killing of the islet grafts? And there's foundational studies showing that that is possible using gene therapy. But that has serious technical and translational limitations, particularly if you're going to be treating children. So other studies have shown that using the protein itself, the presentation of the protein is critical to function. And for the protein to function properly, it needs to be immobilized on a surface like it's on the surface of a cell. So simply injecting the soluble protein is not going to work. Okay, so our work takes advantage of this engineered protein, a recombinant protein that contains a multimeric copies of the Fas ligand protein and is fused to streptavidin. So everything is coupled together in this cluster, and the streptavidin, we can use it to immobilize the protein onto surfaces. Okay, so the strategy that we took is, can we make a biomaterial, a hydrogel particle, to immobilize and present this protein? So for that, we use microfluidic polymerization. As I had described before, we simply use a microfluidic. By controlling the nozzle and the flow rates, we can control the chemical composition and the size of the resulting hydrogel particles. And we can vary the flow rates, and that, as you would, will vary the size of the particle. Again, the idea is to present the Fas ligand on the surface, and you can see that when we engineer the particles to contain the biotin streptavidin binds to, we can immobilize fluorescent particles that have the streptavidin. If you don't include the biotin, there's nothing that gets immobilized, right? And using this system, we can then show that on these particles, we can very precisely control the density, the amount of the therapeutic protein that's on the surface. And very importantly, that protein retains full activity. So as I mentioned, the goal of the protein, what it does is to induce programmed cell death on the target cell. And you can see that we have dose-dependent increases in cell death as we vary the concentration of the surface. Okay. The question is, does that work? So I'm going to show you studies in a preclinical using diabetic mice. So what we did in this study was we took allogeneic islets. So those are islets from a different type of mouse, simply them with our particle that's coated, and then transplanted them in a diabetic mouse that's a different strain, right? So these are different types of mice. This mouse should reject the graft. And that's what we're plotting here, is a percentage of the graft that persists. That means the number of mice that are cured as a function of time after transplantation. Okay, so first the control. So that is the black circles. Those are islets that are transplanted with control on functionalized microgels. And again, this is all without chronic immunosuppression. Okay, so the control group very quickly rejects the graft, right? So within 20 days, all of the grafts have rejected, showing that this is a very stringent model of islet transplantation. If we then look at groups that present islets with the fast ligand microgel, those are the blue triangles. And you can see that we have double the graft time, right? So we push this out so the grafts last longer. And importantly, about 25% of those recipients have long-term graft acceptance. So that was an exciting result and suggested that this approach could work. Now, our collaborators on this project, the immunologists, had previously shown that if you combine the ligand with a course of rapamycin, 2 weeks, you could boost the immunomodulatory effect of fast ligand. And rapamycin is a drug that is currently used in clinic. So from a translational standpoint, that would be an acceptable approach. So that is shown in the red curve, and the results are remarkable, right? Over 92% of those recipients remain immune accepted, right? So they immune accept the graft, they are cured, they have normal blood glucose levels, they continue to gain weight by any metric. These mice have been cured. And importantly, the graft in accepted, but the mice are still immune competent. So that means that they're not suppressed. So if you challenge with a different type of graft or a cold infection or COVID infection, these mice are able to reject the infection while maintaining the graft. We take them out, the days, and then we whether we can still find, the transplanted graft. So you can see that stain for insulin in close-up position to the biomaterial. And then at sacrifice, if we evaluate graft function, and the way this is done is you shoot the mice with a bolus of glucose, right? And what the mouse should do, a healthy mouse that is shown here with the gray lines, takes the bolus and very quickly restores them to the normal level. Because it's stabilizing the glucose by secreting insulin. Graft function, whereas the rejected ones don't. We did a lot of analysis. This was a very exciting result. So we did a lot of analysis in this system that suggested that the way the biomaterial induced graft acceptance was by training the immune system to generate a very important type of immune cell. All of the immune responses in the body. So to demonstrate that indeed the biomaterial induced Tregs, we use a transgenic mouse that allows us to selectively deplete all of the Tregs by injecting a drug. Okay, and then the mouse recovers, but you can see that after injecting this drug, we can selectively delete only the T regulatory cells, leaving all of the other cells in the mouse intact. Repeated the experiment of the transplantation. First the controls, again, you let them go, they continue running, permanently with good survival. And then the experimental treatment, the mice are doing very well. We inject the drug to deplete, and as you would expect, very quickly, all of those grafts reject. Right? So this is a very important mechanistic study that demonstrates that the immune acceptance that the graft generates is due to the generation of T regulatory cells. So in continuing this work, we were very interested in testing this in a large animal model of diabetes. And the most challenging model, and really what's considered the ideal model before testing this in human, is on monkeys, on diabetic non-human primate models. So this is a very challenging model, and we were very fortunate to receive funding for JDRF to test the model. We just completed the study. We treated 4 monkeys, and we were able to cure 4 monkeys, and the monkeys, did as well as we hope. For the duration, for the 6 months that they were transplanted, the 3 controls very quickly rejected the graft. And I want to show you a representative result for one of the monkeys. So this is traces for one monkey. On the left axis are the blood glucose levels. Anything above the hash line is hyperglycemic, so diabetic. Anything below is a normal recipient. And then on the right is the amount of insulin that you need to provide to control the blood glucose levels in the animal. So you can see that prior to transplantation, the animal is very diabetic, requires very high dose of insulin to control the blood glucose. And remarkably, after islet transplantation, very quickly, this monkey became normoglycemic and had excellent blood glucose control over time. And you can see here that the animal is essentially completely off insulin. And finally, to demonstrate that the cure of the animal was due to the graft, we explanted the graft, so took the graft out. And you can see that very quickly, that monkey became hyperglycemic. So these results are very impressive, and our collaborators that did the study shared with me that in 30 years, he's never seen anything that worked this well. So we're very excited about this. This is now getting submitted for publication. But this technology has been licensed to a startup that's working to bring this to a first-in-human study, hopefully within the next year. Okay, so the other 2 stories are quicker. The first one deals with bone infections. So these are, very prevalent and serious cases. In particular, if you look at an open fracture, okay, so this is Devesh running a skateboard, falls down, breaks his bone, and his bone cut through the skin. Okay, that's an open fracture, or somebody in a car accident. So it's estimated that bone fractures, when they're open, about 30% of them become infected, right? So that's a pretty high number. About 2/3 of all bone infections involve bacteria from the Staphylococci species, okay? So those are the target that we want to deal with. And the way this is, when you have an infection, is they in the site and they load the patient with high dose antibiotics, but those therapies have limited, and in many cases lead to implant removal, and that's why this ends up being expensive, right? And part of the reason is that this bacteria end up developing resistance to those antibiotics because the delivery is not efficient, and the bacteria form biofilms, which are communities that are resistant to antibiotics. So there's a clear need for alternative to antibiotics to treat these bone infections. So this provided an opportunity for us to work with a protein. And what you need to know about lysostaphin is that it's an enzyme that has specific activity to the Staphylococcus species, which is what we're targeting, right? And if you look at the structure of the protein, it has a domain that localizes the protein to the surface of the bacteria, and then a catalytic domain that cleaves the cell wall. And when you cleave the cell wall, the bacteria explodes. So it kills the bacteria very, very quickly. One of the things that we like about this enzyme is that it works by different mechanisms than antibiotics do. So potentially you could use it against antibiotic resistant species. So lysostaphin has been tried and shown to be safe, but the problem is on the delivery. Lysostaphin has a very short lifetime. The half-life of lysostaphin is about 15 minutes. So very quickly it falls apart. And there have been problems with burst release from biomaterials on delivering lysostaphin. So this provided a good opportunity for us to engineer biomaterials for the release of lysostaphin. We went back and engineered our synthetic half, in this case for controlled delivery of lysostaphin. So some studies here showing that when you deliver lysostaphin from hydrogels, you can kill Staphylococcus. So you can see that the results are These are different strains of Staph aureus, clinical Islet of epidermidis, and you can see that in all of them, this is the control. You have very high levels of infection. After delivery, or when you treat them with a hydrogel that has lysostaphin, you're reducing, essentially eradicating all of that infection. These are 7 orders of magnitude that you have killed down those bacteria to similar levels as a sterile. And as I mentioned, this is a very potent strategy to treat bacteria in biofilm. These are that antibiotic resistant communities. Again, the bacteria here are labeled green. When you release lysostaphin from the hydrogel at this concentration, we effectively eliminate bacteria. If you look at just the soluble protein, a lot of the bacteria remain. And again, this has to do with the stability of that protein. So we wanted to in an in vivo model. So my lab had developed this model in mice. So this is what's called a critical size bone defect. And what that means is you take out a segment of bone here, 2.5 millimeter. Okay, and BMP2 is a protein that is currently FDA approved for treating bone fractures. Okay, so the model is very simple. We create this defect lab, and then a sleeve that contains the hydrogel and whatever treatment you want in there, the bacteria, and then we monitor the healing and the killing of the bacteria. Okay, so first looking at bacterial counts. If you look at the counts here, this is the infection control, right? So even at 8 weeks after the injury, you have very high level of bacterial presence at that defect. This is a nasty bacteria. This is isolated from a bone infection in a patient. So you have very high levels of bacteria. If you treat instead with a lysostaphin containing hydrogel, you essentially knock down all of those bacteria. And at 8 weeks, there's no differences. You're actually below the detection of the assay between an infected defect treated with the lysostaphin hydrogel and the sterile control. Importantly, we wanted to benchmark this against the current clinical care, which are injection with antibiotics, in this case, gentamicin, and you can see that, if you treat disinfected defects with gentamicin, there's no reduction in bacteria. So we can kill the bacteria, but can the bone heal, right? So we repeated the experiment and then assess formation of the bone healing. So this is the defect here, and we do micro CT, so essentially X-ray, to quantify the amount of bone formation in the different defects as a function of time. So that's summarized here. Again, if you look at the amount of bone volume in those defects, for the infected case, very low amounts of bone. The ones that you treat the infection with lysostaphin hydrogel, you increase significantly the amount of bone that forms in the defect, and the levels of bone formation are statistically equivalent to a sterile defect. Okay, so we've eliminated the bacteria, able to produce bone level as a sterile defect. And again, if you treat with the standard of care, which would be the antibiotic, those defects remain unhealed. And finally, we wanted to test the quality of that repaired tissue by measuring the mechanical, right? So we do a simple torsion test of that repaired tissue. As you would expect, the infected defects that have no bone have very poor mechanical integrity, right? They fall apart. The ones treated, the infections treated with the lysostaphin have good levels of mechanical strength, and they're equivalent to the sterile implant and approximate the levels. The red line are the levels from a bone that was never fractured. Okay, so we're getting very good reconstitution of the mechanical properties of that repaired tissue. And as you would expect, the ones treated with the antibiotic doesn't work. I'm going to this in work bringing this hopefully to one day treat patients. We've completed a pilot study, in this case, a sheep model of infection. So you can see one of the sheep there. These sheep are huge. They're about 200 pounds. I had to get in the cage with the sheep to get him out into the table because he didn't want to go. So, large animal model. The model that we're doing is a bilateral screw on plate on one of the bones in the leg. So what we do is we put a plate with two. So we took out this to 1 week, and you can see the results here in the surrounding tissue or on the hardware, right? The plate on the screws. And you can see that remarkably, when we treat with the Lysostaphin hydrogel, we significantly reduce bacterial counts. This is on a log scale to eradicate that infection. So these results are very promising, and this is a continued study to see if we can develop these anti-infection materials. The last story I'm going to share with you, I'm going to go quickly, but I'm very proud of this story because when I wrote the grant proposal, it got trashed. I mean, the reviewers hated it. They said it was never was going to happen. I think it's a good story for the trainees in that if you have a good idea, you should persevere and you should try it. This is also a fantastic collaboration with first range of make this project really a success. So the work deals with stem cells. So these are human stem derived organoids. And this is a very, very technology that was developed a few years ago, not by my group. Many groups use this now. But what people notice is that you can culture human stem cells in the lab, in vitro. And if you culture them under the right conditions, will assemble into multicellular complex three-dimensional tissues that have properties to the adult tissues. So people grow in the lab this what are called human organoids, and you can do them for intestine. Those are called HIOs. Those are the ones that I'll present data on. You can do lung, kidney. So you can imagine this is really a transformative platform for development and disease models because you can generate tissues like brain tissue from human sources and from human patients. So for example, for Alzheimer's, there's really not very good mouse models of Alzheimer's. But you could take the blood cells from an Alzheimer's patient, reprogram them into a pluripotent stem cells, and then generate human organoids, and then use those for drug screening. Another application that this crazy guy at Emory does is he uses human organoids to study Zika infection. And you can't do that study in a mouse because the mouse doesn't get infected by Zika, right? So that's an example there where this technology allows those researchers to do experiments that you couldn't do otherwise. So as these organoids can be derived from either human embryonic stem cells or induced pluripotent stem cells. And for many of these tissues, you have to culture the cells in Matrigel. Okay, Matrigel is a commercial product, but it's a heterogeneous mixture of proteoglycans and growth factors. The way Matrigel is prepared is that you have these that lay down a very complex matrix. There's over 1,000 different components in Matrigel, and the company extracts that membrane, gets rid of the cells, and sells you that extract to use. So many groups use Matrigel, and there are really two major limitations to use of Matrigel. First of all, because it's so complex and you're seeing different cell lots, there's a lot, significant lot-to-lot compositional and structural variability. So if you're on Matrigel, you got to screen a lot of lots to find that works. Brought lots out, you got to do it again. And more importantly, the use of Matrigel has limited clinical translational potential because Matrigel is from mouse tumor cells, right? So if you grow human organoids and wanted to use it for a therapy in humans, Matrigel limits that because it's from a xenogenetic tumor source. So in this project, we asked whether we could engineer a synthetic substitute for Matrigel to grow organoids. And I'm happy to share with you that we did, and we had the cover of this great cell biology journal. So simply the way you generate the organoids is that you culture your cells in a planar surface, and under the right conditions, about day 5 or day 6 in culture, the cells bud off into clusters, okay? And these cell clusters bud off from the surface into the media, okay? Those are called spheroids. And what people in the community do is they collect the spheroids and then embed them in Matrigel. And when they're in Matrigel, they end up growing into the organoids, right? So we did the same thing, collected spheroids in our synthetic gel or Matrigel. And we wanted to see what formulation, what biophysical and biochemical characteristics of the synthetic system could recapitulate what you see in this very complex matrix that is Matrigel. So I'm going to show you some results here in which we fix the biochemical properties of our synthetic gels. So they have the same, all of these gels have the same density of an adhesive peptide and a degradable cross-linker. And simply what we vary was the mechanical properties of the gel. You can't do this experiment with a biological system. You really need to use a synthetic system. So you can see the spheroids here. And then if you look in the lot with stain, you can see that they're alive in this 2 formula at 2 hours, but on the stiffer gels, all of them die. Okay, so to maintain the cells alive, you need the softer formulations, about 100 Pascal of the storage modulus. And if you continue culturing those, these spheroids will grow into a structure that looks like an organoid. And when you stain for it, they form lumens and have the epithelialization that you would expect in an organoid. And we really didn't see any difference between the organoids that were grown in the synthetic system compared to those in Matrigel. Now, to really get full differentiation of these organoids, what typically you do is you in an animal, and then with the in vivo juices, they fully differentiate. So, these crazy journals make you do this thing. So we went ahead and did that and generated organoids either in the synthetic system or in Matrigel, implanted. And many of you may not know but what testing looks like, but you basically have these structures that form villi, which look like fingers. And you can see those beautiful structures here and down here. And the important thing is that you don't see any difference between the human tissue generated in the hydrogel for the organoid in the Matrigel. And if we look at any of these markers, there's really no difference with it between the organoids that were made in the gel and those that were made in Matrigel. So that's a significant advance because we can now make organoids in the lab that don't use a Matrigel, and you're using a fully synthetic system. We wanted to take that a step further and wanted to see whether we could use those organoids then for a therapeutic approach. Okay, up to this day, nobody has used the organoids in a therapeutic application. Okay, so for this study, we use a model in which we generate mucosal wounds in the large intestine of immunocompromised mice using a colonoscope. It's done by our collaborators. We don't know how to do this. So what we do is at day 0, we have forceps and we can generate a wound in the intestine of the mouse. And then we come back at day 1. This is all under fiber optic. We can come at day 1, we find the wound, and then underneath the wound, we inject whatever treatment we want to. We come back at day 5 and then measure how the wound goes. And you can representative traces here from day 1 to day 5, right? So if we look at the results of the wound closure, this is no injection. About 50% of the wound has closed at 5 days. If you inject hydrogel by itself, there's no difference. If you inject organoids by themselves, no difference, right? So there's no advantage, there's no treatment there. But interestingly, if you organoids that were grown in the system with the hydrogel, right? Because what we're doing here is we can inject them using our biomaterial carrier. We see about a 50% increase in the wound closure. So the wounds heal much more faster. So this is the first example of a human organoid in a therapeutic application, and it's mediated by using the hydrogel as a delivery vehicle. Now, 5 days, there are many reasons, or there are several reasons why this would give you better healing, right? It could be that the organoids go in, dump some regenerative factors, and then die. So we were very interested to see whether the human cells will engraft into the mouse intestine. So we repeated the experiment and then look at engraftment of the human cells at 4 weeks. And in this preparation, the human cells stain green, the mouse cells don't stain. And anything that blue is all cells, mouse and human. When each of the wound, you can see a clear transition from mouse tissue to tissue that starts having human cells as part of the tissue. Remember, this looks like fingers sticking up. And remarkably, when you go to the center of the defect, the majority of that tissue is of human origin. And you can see there the beautiful intestinal villi. And that only happens when you deliver the hydrogel, when you deliver the organoids with the hydrogel. Functional, right? They can poop. That's a very important functional measure. It means that you don't have adhesions in the intestine. And at the time, this was the first application that you could get human organoids to engraft in of colonial injury. So this work is now expanded, and we're looking at applying it to intestinal wounds under inflammatory conditions like Crohn's disease or inflammatory disease. So with that, I want to thank you. Just to summarize, I told you 3 things demonstrated how we can use this hydrogel platform technology for delivery of therapeutic cells and proteins. I told you about transplantation for type 1 diabetes. One of the things that is really exciting about this concept is the off-the-shelf application, right? We can make the material in one site and ship it to be transplanted at the time of the surgery. I told you about the anti-infection hydrogels. We've shown that that works in different applications and that it's effective against antibiotic bacteria. And finally, I told you about using the synthetic system as analogs of natural matrices for the development and therapeutic applications of organs. So I want to thank you for your time. I acknowledge the sources of funding are great collaborators, and just give a shout out to my past and former trainees, particularly the ones that did the work. Deb was a very talented PhD student that started the fast ligand work that was finished and continues in the monkey with Maria Coronel, who's really a phenomenal postdoc in the lab. Is an MD PhD student. He's now doing his residency in orthopedic surgery. He started the bone infection project. Now that is by Jeremy Kaplan, who's done the sheets, and Pranav Kar, who's a postdoc doing infection. And then finally, the organoid studies were started by Ricardo Cruz Acuña, great PhD student, now a postdoc at Columbia, being continued and expanded to the inflammatory model by Adriana Mulero-Ruz, who's also an outstanding student. So with that, I'm sorry I went a little long, but I'd be happy to take any questions. And I'm sure this is not the typical mechanical engineering lecture that you get on Fridays.
Excellent. Thank you so much, Andres. So I am trying to figure out yet another web platform. So if anyone would like to raise their hand, that is fine, or you can just unmute yourself and ask Dr. Garcia a question.
Hi, Joanna. This is Siddhartha. I have a few questions.
Fire away.
Can you hear me?
Yes.
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 Wonderful talk. Thank you so much. This was so much revealing. So actually, I can tell you one thing that you showed that I used to have as writing in the part of the introduction for the soft wetting papers that we wrote. So we used to study the dynamics of drops, liquid drops on gels. So one motivation for ours, like to introduce the topic, was a very famous paper by Francois Brochard-Wiart, a French scientist. She did a lot of work with a very senior scientist, very old scientist. So she once showed in a paper published in Soft Matter that if you have a rigid substrate, tumorous cells spread. On the other hand, if you have a gel, they don't spread. This is something very similar that you discussed where during your organoid talk, where you showed that if you have a soft gel-like thing, the cells don't spread and they just accumulate and form the structure. So my question to you is that two questions to this. So is this kind of interaction specific on the cells? I mean, like if you change the cell or if you change the nature of the hydrogel, does this kind of arrest of spreading of the spreading away of the cells, do they get arrested?
Yeah. So, you know, that's a very good question. So that's in the area of what we call mechanotransduction, and certainly the mechanical properties of the surrounding matrix of the cells will influence the responses. And as you indicated, there are permissive formulations that allow spreading, and there are non-permissive formulations. Now, the machinery seems to be conserved for different cell systems, but the details will vary for different cell systems because, for example, one class of cells may express different levels of one receptor or may express a different contractile protein than others. So I'll give you an example on the organoids. On the same material, on the same formulation, we can grow intestinal organoids and organoids that look the same, as you would expect them to do. But if you try growing a pancreatic islet organoid, it will not grow in that formulation. You have to find a different formulation, right? And what makes them different often is the biochemical details of the matrix.
Okay, so is there a follow-up to this? And this is something that I'm personally very interested to understand. Are there any thorough theoretical or computational kind of a template that, okay, here is the kind of gel properties that we desire for these classes of cells to ensure that we form organoids?
Yeah, I mean, there was this theory of the interfacial energies that was developed, you know, by Weinberg, and you know, there's an underlying theory, and if you're interested, you know, send me an email. I can dig up the papers.
Yes.
But they basically had characteristics in which cell clusters will segregate or come together. So there is some biophysics driving some of this process. It's just that it's not a universal phenomena, or the different cell types will change the parameters of the model.
Okay, good. Yeah, that would be it from me. Thank you. I might ask one more question if everybody. I would like to give everybody a chance. Yeah. I see one. Yeah.
Yeah, there's one question in the chat. I don't know if Encore, do you want to unmute?
Yeah, Andrés, great talk. Question is that can you comment more on the rapamycin study in the Islet part of the talk? So is rapamycin locally delivered in these cases, or is it systemically delivered? Because if so, then a whole bunch of cells are going to shut down their mTOR system, and what could be the consequence of that?
Yeah, so Encore, in this model, and that's both for the mouse and the monkey, the rapamycin was delivered systemically. And the reason for that is, you know, those are the general protocols that are typically implemented. Now, certainly, as you point out, you would expect a systemic effect. I will point out that the controls, particularly in the monkey study, The controls had monotherapy of rapamycin, and they still reject it, right? So you do need the fast ligand for it to have graft acceptance.
Right.
And remember, from a translational regulatory standpoint, because the rapamycin is currently delivered systemic, we want to keep that because that's going to be a lower barrier to the FDA than to engineer a material that also releases rapamycin locally.
Yeah. Okay. Thank you.
Mm-hmm.
So Andres, great talk. This is not my field, but I was going to ask something. I always felt fluid mechanics was colorful, but I saw a lot of color images. So what technique do you use to visualize these cells? And what's the limitation of these technique?
So it depends on the application, right? If you want to look, let me, so Devesh, are you asking,
So
the chat. So the picture with all the colors, right?
correct. Yeah. Yes.
Like this.
Yeah. Like, let's go on
Okay.
this one.
Yeah. So for this, what you do is you have an antibody that has been generated to recognize a protein in it. So, you know, you can see there's the color here refers to the target of that antibody. And you can buy antibodies with different colors, and then using confocal microscopy, you can segregate the colors and you can look at them at the same time like that. So typically, you know, in our setup, we can look at 4 to 5 colors at the same time. We also use other techniques that are not using a microscope, but are using what's called a flow cytometer, which is all enabled by engineers because it's a microfluidic system that you flow one cell at a time. There you can do 12 colors at a time. And just to put in a plug for the IBB core facility, we just purchased an instrument that 40 channels at the same time using heavy atoms and a mass spectrometer. So instead of colors, we use heavy atoms. So these are standard multiplexing technologies that all exploit antibody specificity for the target.
And each color, if you look at the gradient in color, so they mean something with the amount of these antibodies. Is that?
If you do the so if you do the experiment right, which is not trivial, the intensity of the color corresponds with the amount of target. So you could use it in a quantitative fashion. The flow cytometry system that I described, you can quantitative pretty straightforward because you're basically flowing one cell at a time through a channel, so all the other parameters are the same. In a section like I'm showing on the screen, that's a little more challenging because there's some effects from the preparation of the section. I don't want to get too technical, but there are ways to do it on something like this. Is a little more challenging, and that can be done to quantify those levels.
Okay. Thank you, Andres.
This is why, you often ask me, why are these bioengineering grants so expensive? Is, one of those antibodies is $500, just the antibody, right? Somebody's making some serious cash out of this.
Andres, I have one more question. This is Ankur. So the gut system, is it expected? There are some reports of how the brain system regulates the pulsatile nature of gut. Is that something that you could model? Is that something that is relevant here, or?
So do you mean modeling it in vitro?
In vitro, yes.
Yeah. I mean, so there are groups that are building these organ systems on a chip, grow, let's say, a organoid and a hard organoid, and you can see how they talk to each other. So what Ankur is referring to is many of these systems have nerve endings in there that will control, let's say, the movement of the intestine. Could you engineer those in the lab to study how drugs affect that, or infection, or we're not actively studying, but you could could do that. And I would suspect, we didn't specifically look at it, but I would imagine that within the diversity of the cells that the organoid generates, there would be cells that could form junctions with nerves. So I think you could do that. It's not something we're doing, but.
Okay.
So Andres, let me go ahead and ask one more question. I think you alluded on your slide before. So there are junior faculty who are trying to get into these areas. And it looks like it's a field which requires a lot of partnership with different groups to really make an impactful work. So what will be your advice for a junior faculty who is starting in this area in a school which might not have all the partners in place?
Yeah, Devesh, that's really a very insightful question. Certainly, and Joanna and Kur and others can certainly comment on this. These fields are extremely interdisciplinary, and there's not one lab that can master all of the necessary assays, techniques, knowledge. You rely a lot on collaborative work with different experts, and I think my strategy with my trainees has been to get them very involved in those collaborations from the beginning, because as the faculty here all know, it's very hard to collaborate. There's you sort of have to have that personal match, and you have to be able to get along. So for a junior faculty, and this is a strategy that I took on when I started, right? Remember, my background's mechanical engineering, this stuff on the animal surgery until I got to Georgia Tech. I would meet with the particular experts and try to understand where are the problems, where are the patients. And I think if you look at the 3 stories that I presented today, we work on a problem that the biomaterial can solve, right? So I thought diabetes study, up because conference, the immunologist came to me at the lunch line and said, do you think you can make a biomaterial to present this protein? And you never ask an engineer, do you think you can do this? Especially an engineer from Georgia Tech, because we're going to say, yeah, we can do that, right? So it was about learning and having that conversation. So one of the things that I love about this field is, there's never a dull moment, right? There's always something to be learned, and you can always generate and establish a conversation with that. So, having a conversation, grabbing a coffee, beer in non-COVID time, one of the challenges of COVID is being able to do that. That's important. The other advice is, I found over the years that going to interdisciplinary conferences, okay, and for me, the favorite are the Gordon Research Conferences, which are small conferences, right? They're 200 to 300 people, single session. Everybody's stuck together in the same prep school in New England with the mosquitoes, biting you. I think those are very conducive to generate potential collaborations. And then I would say, don't be afraid to ask questions, right? I mean, I ask the stupidest questions. I'll tell you a quick question. Do I have time to tell them a quick story?
Yes. Yes.
Okay, so on the JDR, on the diabetes model, right? I presented the data on the mouse, which we work with the immunologists at a conference for JDR, and the program officer comes to me after my presentation and says, "Wow, your data looks fantastic. I would be interested in funding a study in human," right? So I'm stunned. I never had somebody come and tell me, "Give you money," right? Large animal study. So he said, the only thing I ask is that you work with somebody that's here that does the monkey study. So behind him was the person we work with that said, "Oh, we'll be to work with you," right? So we are writing the white paper to ask for the budget. He sends me this budget that is just, jaw-dropping, right? The study, to do the monkeys, unbelievably expensive, right? The monkeys eat better than I do. So I I'm looking at the budget very carefully because I thought this foundation is going to say, no way, we're not funding this. And I see there that there's a line that says we're going to train the monkeys for 3 months before we make them diabetic and put the islets. Or yeah, right, before we make them diabetic and put the monkey charge are $20 a day for the cage. Okay, so I'm doing the in my head, and I said, what are we training these monkeys to do for 3 days, for 3 months before we even do the therapy, right? We're just burning cash. So I very stupidly sent an email, but I sent an email to my collaborator. I'm like, Jim, what are you training these monkeys to do, right? And he sends me a video clip and says, take a look at this and let me know if you have any questions. Okay, so I hit the video, and it's a video of the monkey on the cage, and the animal technician comes and hands the monkey a treat, and the monkey sticks the paw out so the technician can blood without anesthetizing or restraining the monkey, right? And that's something that we would never do in a mouse or a rat. You clamp them down, you restrain the animal, and you draw blood. But on a, you can't do that. Monkey will go literally ape shit and will bite you and will kick you. So, I think it's very cool, right? I think, you have to be, you can't be afraid, right? It's part of a dialogue. I tell you, my life science collaborators have asked the dumbest questions about the engineering side, right? So it's just a matter of having that relationship.
Thank you, Andres. So I think it's almost 4:45, so I'm going to go ahead and thank both you and Jonah. Jonah, thank you very much for taking time and being here and moderating this session.
Thank you
Thank
for the
you.
invitation, and everybody stay safe.
Thank you.
Yeah.
And Corey, you can just call me on my phone and we can talk.
Yeah, I
Take
have
care.
some suggestions. I'll call. Bye.
Yeah, sure.